everyone. Again, welcome to today's webinar. I'm glad that all of you have tuned in. So today we'll try to see the complete introduction of PyMol. How can we use PyMol for our most of the activities that we do, and how are we employing PyMol in order to make images for publications. This webinar will give you a hands-on onto most of the basic operations that we perform using PyMol, and it will introduce you to the basics of various functions that can be done using this. So this is the complete navigation for today's webinar. Initially, we shall go ahead with the introduction and how do we search for structural information using online platforms, and how do we obtain structural information from the files that we download for visualizing PyMol. After that, there'll be an introduction to the GUI interface as well as the command line interface of PyMol, followed by which we'll have hands-on on performing some of the key functions using PyMol. And after that, we'll wrap the session by understanding how can PyMol be used for making images for the publications, how are figures for publications done using PyMol, what are the commands that we need to understand. So this will be the basic outline for today's webinar. And without any further ado again, let me go directly into the basics of PyMol. So PyMol is an open source software that's introduced by Schrodinger. So it mostly helps us in visualizing the biomacromolecules, for instance, the DNA, the protein, and ligands. And it helps us also do various other functions, like for example, understand the interactions between different biomacromolecules or find the binding pockets, and also make these visualizations into live figures which can be used for various purposes, like for instance, the publication. As you're seeing my screen right now, we are into the home page of PyMol, that's by Schrodinger. So this is where we download the open source software from. For downloading, we have multiple options where we can download it for various OS options, like for instance, the Windows, Mac OS, as well as the Linux. So downloading is quite simple. We have an executable file that gets downloaded, and double clicking on the executable file will help us unpack the library that we're downloading, and it will immediately install the software for it. And for today's webinar, let's primarily focus on 2-3 example structures, and let's see how can we use these structures to understand the structural information and how can we go about their visualization. So for today, the first example that I would be taking is one particular structure whose PDB ID is 3UZV. So 3UZV is the structure for the D3 domain of the dengue virus, which is in complex with the antibody, a structural antibody, which has been raised specifically against it. So in order to download the file, so we click on download files option in the PDB database. We click on the PDB format. So this would give a prompt for us to download the structure. So we have already the structure downloaded here. However, for your information, I'm downloading it again. And when we go to visualize it here using the file manager, I think you can see the file that has been downloaded over here. Before going into the structural aspects which we visualize using PyMol, let me just introduce to the text format of what this file is. So I'm opening this file that has been downloaded using a text editor. So in the text editor, I think you can see a lot of information. Usually any structural file, any coordinate file which we try to visualize using PD with PyMol would have a lot of information, which includes the necessary information as well as the supplementary information. Like for instance, here what you see on the top is all the structural information, the supplementary information. For instance, the header of this file, when was this structure deposited in the PDB database, what's the title of this structure, what is the name of the paper, who are the authors that are being affiliated with this particular paper. All this supplementary information is being deposited onto this file. As we scroll down a little bit, we see a particular line where we have this key term named atom. So this is exactly where our structural information begins. In order to have any structural information to be visualized using a platform such as PyMol, we need one important information, that is the coordinate information for any particular biomacromolecule. So in this case, if we are trying to see the coordinates in order to visualize the 3D molecule, we particularly need 3 important coordinates: the X coordinate, the Y coordinate, as well as the Z coordinate. With these 3 particular coordinates, any atom can be represented on a 3D space. So that way, all the atoms that are involved in this particular protein structure have been shown here using three different coordinates, the X, Y, and the Z coordinates. And this can be visualized here. Which particular atom is this has been shown on the extreme right, whether it's a carbon, whether it's a nitrogen, whether it's an oxygen, all this information has been given. So as far as visualization is considered, the most important aspect would be this coordinate information, and rest of the supplementary information, all of this has been removed from the file. This can still help us visualize the structure. And the predominant protein that we are trying to see has been represented as atom. The keyword for representing the protein structure is the atom. Whereas apart from the protein, we also have other secondary atoms, which can be the ligand, the water molecules. So these atoms are represented as heteroatoms, which you can see using this particular key term, which is called the het atom. So heteroatom again represents the secondary atoms, like for instance, water, ligand, or any other metal ions that are bound to the protein. All the PDB files, all the coordinate files end using this particular key term called end. END is the key term that helps in understanding that this is the end for any particular structural coordinate file that we are trying to visualize. So now So now I'll go ahead and open this particular file using PyMol. Again, before going to PyMol, as I was saying, we have two different versions of PyMol. As you can see here in my screen, I have two different versions of PyMol being installed. One is the normal PyMol version, which we get to download, and it's for using specifically for various conditions using a license. Now, for instance, the software that is being opened currently is the normal version of PyMol, which is for corporate purposes where we use a license file. And you can see here that currently there is no license file being activated. So using a particular license file which has to be purchased, we can run this particular version of PyMol. Whereas the second version of PyMol is for academic purposes, which is an open source software. You can see it is being mentioned right on the top. The open source PyMol helps us to use PyMol for various institutional and academic purposes. And the other important advantage of using an open source PyMol is it helps us design various figures at highest resolution possible, which we'll be looking at in the near future in this webinar. How to design high quality images, high resolution images for publications that can be achieved using PyMol open source. Whereas in order to generate high quality images or the high resolution images using the normal version of PyMol, we might need a license file that is missing currently in this version of PyMol. Let's go ahead with the file that we had downloaded from PDB, whose PDB ID is 3UZV. So right now, let's click on open with PyMol. I'm opening the structural file, the coordinate file that I downloaded using PyMol. Uh, so the file is being opened. So as you can see, by default, we are seeing this protein file in particular conformation. What conformation is this? What other conformations can be seen? What are the different types of representations with which we will be able to visualize this protein? Let us see in the next part. So right now, you can see this protein molecule, which we have opened, the exact same thing that we saw in the text editor. So now, how do we see what particular protein molecule this is? It has loaded as 3UZV, the same file which we had saved in the text editor. And now, in order to see the sequence of this particular protein, if you can see on the right hand side, there is a small button named Yes. So I'm clicking on that, which helps me get an idea about what is the sequence information of this particular protein. So on top, you can see the sequence information that is being displayed here. So as I go on selecting them, all the particular residues corresponding to the structure are getting selected. So this is how I have loaded the file that was being downloaded onto my PyMol software. So again, giving a brief introduction about the interface, the overall interface of PyMol that you're seeing here. We have two important the operations that are available. One is using the command line that you can see here. There is a command line option seen, and also there's another command prompt that you can see on the bottom side, and on the top side you have another command prompt. In order to use using the graphical interface, we have another operational facility called the GUI, which for short is the graphical user interface. which you can see on the right side. Please follow the cursor, the mouse cursor that you can see on the screen. I'll try to explain how the entire interface looks. So we have two different visualization state. One is the command line based action where we can feed in particular actions to the command to the PyMol software. And on the extreme right, we have the GUI, which is the graphic based interface. Now, for instance, if I need to perform any particular action onto my PyMol structure, then what I do using the graphical interface is here we have an interface where we have various buttons. Like for instance, we have a button for action, we have a button for showing any of the operations, we have a button here for hiding any particular operation, we have a operation for labeling, and we have a button for the color command. So these are different actions that we can control using the graphical user interface. And we also have an interface on the extreme right bottom that you can see here. Most of these details are being shown. We have multiple buttons in order to control the functions that we want to do. And on the other hand, if I go onto the extreme left, we have a command line where, for instance, I can type any command. You can see, for instance, I have typed the word command, and this is taking as a text-based command. So any of these particular commands. For example, here, what can be given as one particular function in the GUI can be given in the text format in this command line prompt as well. And the same can also be given on the extreme bottom that you can see on the left-hand side. So whatever I type is getting typed onto the bottom. So these are two different interfaces with which I can communicate with my PyMol software. The first one, again, I'm repeating it for everyone. The first one being the command line interface, where we type in a particular command in a text format in order to communicate with PyMol. And the second one being the graphical interface, which we have various buttons for. We choose these particular functions in order to communicate with PyMol. So on the extreme top, the left top side of my PyMol window, you can see multiple functions that are present here. So let me go by explaining each of these particular functions one by one. On the extreme left, you have this option named file, where we control the type of file that we are opening, whether we are opening a coordinate file, whether we try to save this entire visualization state in any particular format, or whether we are trying to run any of these functions in PyMol using a particular script. All these functions, any file-based functions in this particular PyMol interface can be controlled using this particular function, which is file. What all are the significance of these options that you see here, we will see in the later part. In order to go for building any particular molecule, like for instance, if I'll have to build a particular residue or a particular chain onto my macromolecule, I would use this build option. In order to create any movies using PyMol, which will be an animation or an MP4 file where we are trying to see a movement in these particular structural components, we use the movie option that's being given here. And in order to go about selecting different visualization state. Now, like I told in the beginning, what the protein molecule that has been loaded in this window, we have it in a particular visualization state. Apart from this visualization state that we see here, we also have various other visualization state. How do we control all these visualization state? We can change using this display option and various other settings, like for instance the transparency of or the different visualization state. What are their secondary options that can be changed when we are visualizing a particular biomolecule using PyMol? All those settings can be changed using this particular function called the setting. And probably whenever we are controlling the PyMol interface, when we are trying to visualize the biomacromolecule, we use our mouse for performing various functions apart from the keyboard. So a single mouse, the multiple buttons that are present in your mouse can be helpful in doing various functions. So in your fingertips, you have the option to control multiple options at the at simultaneous point. So how do we control what button of the mouse helps in performing a particular function? This can be altered using the mouse function that you can see here. And in order to go for some higher calculations, like for instance, if you want to create a mutation in the protein structure, or if you want to calculate the atomic distances between two different atoms in my protein structure, all these active actions can be performed using the wizard option that you can see here. And apart from that, PyMol is a software that is given to us by Schrodinger, but there are various codes written by other people in order to enhance the activities of PyMol. Because PyMol, as a mere visualization software, has limitations of its own. It is bound to certain functions. But how do we enhance these functions in order to perform activities that we want to do that PyMol doesn't offer? For this, people have written various codes, people have written various programs. These are the third-party programs that can be installed as plugins into the PyMol software. So how do we install and how do we use them in order to do the desired function? That is taken care of the plugin option that you see here. And the final tab that you see here is the help option, which in order to receive various helps, like for instance, if you're stuck at any particular point, if you would want to understand what does a particular function do, or what is the action that is invoked by a particular key term that you enter in the command line. These particular helps can be obtained by using this particular tab that you see here. Now let us start exploring each of these tabs in specific right now. So let us start with the file option that you see here. In order to open any particular file, any 3D visualization state using PyMol window, you can use this open function. For instance, if you saw a while ago, we double clicked on this particular coordinate file, which we had downloaded from the PDB database. So we double clicked on this, so it directly loaded onto the PyMol window. But in order to open it directly from a PyMol window, I would go about opening the PyMol window at the beginning. And right now you can see that no structure has been loaded into this. If I will have to load this particular structure, I can do that by clicking on the file option, open, and then I will go to this particular folder where my file is present. I will copy the path for this folder from the file explorer. I would go to that particular folder, and you can see the file that was downloaded previously is visible right now. So I will click on this and click on open option, so that this particular biomacromolecule that was being downloaded from the database, PDB database, has now been loaded into the PyMol window. And using mouse, you can control various operations that you can see here. So this is one option where you can load. And in order to save a particular session, for example, let us say that you would perform a particular function, which you'll be seeing in the later on part. If we perform any particular actions onto this PyMol window, that particular session can be saved using the save session option, where it would be saved as a PyMol session file, which at any particular point can be saved and visualized, so that we pick on from the point which we left at the last particular working state. So that is one particular thing. And I was telling previously, we can also run a particular command using the script option. For example, any particular script in that has to be invoked in a PyMol software can be saved as a PML file. So PML is the extension in which we save the files. If I go to the file again, the PyMol file that was being the 3D coordinate file that was being downloaded from PDB has this particular extension called the protein data bank file. Whereas a PyMol session file that was being saved from a PyMol window has this extension called PyMol session file, which is saved as a PMol file. And at the same time, in order for me to open any particular script, I can save the file in PML format. Now, a PML format is nothing but a bunch of commands that we would want to run in the PyMol window, which again we will be seeing soon when we go to the command line prompt. So now let us open a particular structure for which we'll be opening the PyMol window. And I would click on File and Open, and I would click on 3UCB structure that I have downloaded from the PDB database. So now this loads my structure into the PyMol window. Now here, as I was mentioning previously, this helps us in controlling the graphical user interface for PyMol. We have various buttons. Like for instance, let me explain each of these buttons. This particular button that you see here, which is being titled as A, stands for action. where you have multiple options that are used for assigning different operations into my biomacromolecule. And after this, we have a button named show. If you want to display a particular representation style or a visualization state to the macromolecule that we have loaded on PyMol, we would click on this option show. Like for instance, right now, the visualization state that is open in PyMol is called the cartoon state. If you could click on show, and if you see, there is an option named cartoon. So this is the default visualization state that is present for any biomacromolecule. And for instance, if I would want to represent this using a different visualization state, for example, right now, let me go ahead with this representation style called the surface. Once I click on this option called surface, you can see how the visualization state changes to a different format. Now, this visualization state in PyMol is called the surface. Or for instance, now if I would want to hide this particular state that I have showed, we have a third button called the hide, which is represented using a H button. So now if I want to particularly hide the surface option that I had shown previously, I click on surface and that part is now getting hidden. Now, right now, again, what we see is the default option, which is the cartoon. And if I would want to hide the cartoon visualization state as well, I again go to hide option and then I would click on cartoon and this would hide my cartoon state as well. So this is how we are efficiently going to use the show and the hide buttons in order to visualize different states for my macromolecule that I have downloaded. Let me again go to show option and I'll click on the cartoon option. So we are again seeing the default option. So after these three buttons, we have this fourth button called the label. I think you might have seen in various publications, various papers where they try to label a particular number of the residue or the name of the residue or any particular notation for the ligands that are being bound to these proteins. Any sort of labeling which is a text-based labeling that has to be done in this window can be done using this label button. For example, if you would want to label the residue name, you would click on the residue. If you would want to label the residue using a single letter for each of these amino acids, you would click on the residue one letter option, or if you want to say, or if you want to label the chains that are present in this particular structure, you would click on the chain and so on. We have various labeling states for multiple options that can be seen in PyMol window. And finally, as we're talking about the visualization state, we see the molecule being loaded in a particular color. In order to change different coloring patterns, the coloring schemes for these biomacromolecules that we visualize using PyMol, we have the fifth button called the color, so which is represented using the C button over here, the last button. So I click on that and I see options to label these particular biomacromolecules using different coloring schemes. Right now, what has been loaded is using a green coloring scheme. If I would want to change the entire coloring scheme to yellow, I would click on yellow and you can see how the visualization state of this entire molecule has been changed to yellow color. So these are the five important buttons that you see here in the GUI or the graphical user interface in order to communicate with PyMon. So let us go by exploring each of these buttons now using the particular functions that they do. For which let me close this entire window and let me load this protein again using PyMol. So let us right now start off with the show button that I was talking previously. Now show button helps us to show various options that are being visualized in the PyMol software as well as the different visualization patterns. Like some of the key patterns that you can see are the line confirmation, the sticks confirmation, the cartoon confirmation, the spheres and the surface. Now let me go ahead by showing each of these visualization states. How do they look? Let me hide the previous confirmation that's seen, which is the cartoon. So let me show the entire biomacromolecule using the stick confirmation. Here you can see that the entire protein that was being loaded here in my PyMol window has now loaded in a particular visualization state called the stick confirmation. As I zoom in, I can see different atoms of all the residues of this protein being shown in particular stick confirmation. Now, this being one of the confirmation, I would hide this now. And now I would click on show and then I would click on the ribbon confirmation. So ribbon conformation is also one of the visualization state where we can try to see the protein in its backbone conformation. So all the backbone atoms, which are technically called the C alpha atoms of any particular protein. So each of the C alpha atoms, which constitute the backbone of any particular protein or the DNA, are being shown using this conformation visualization state called the ribbon state. So now I would go ahead and hide the ribbon state and I would try to visualize the cartoon. Cartoon structure, the cartoon visualization state predominantly helps us to visualize the secondary structure of my protein. I think by now we all are familiar with various secondary structure conformations of a protein. For instance, the alpha helix structures, the beta strands, the loop structures, all of these particular secondary conformations of a protein can be visualized using the cartoon structure. For example, if you see here, all the beta strands are being shown using this particular conformation where we have an arrow which ends with an arrowhead showing the direction of this particular beta strand secondary structure. And on the other hand, all the helices are being shown using a helical structure. Probably we have a very fewer helical structures. Here, if you can see, the helix is being shown using a helical turn. So this is another secondary structure which can be visualized using the cartoon visualization apparatus. And now we are going to hide this particular visualization state, the cartoon state, and we would go ahead with showing the mesh conformation. Now this mesh visualization state helps us visualize the biomacromolecule using the van der Waals radii. Now, what is van der Waals radii? If we look at a physics point of view, each of the surface of the biomacromolecule is being covered by something called as the van der Waals radii, which represents the atomic cloud that is present on your particular surface, the protein, which is constituted by the nucleus and the electrons that constitute your entire atom. Now, each of these, the electron cloud denotes what is the particular van der Waals radii or the shape. To put it in simple words, you primarily see these blobs which represent the whole area that the protein molecule covers in the 3D space. So this entire thing is shown in a mesh conformation. So this is exactly what you see in this particular conformation. So now again, let us hide this particular conformation, which is the mesh. And this last conformation, the visualization state that we would want to see here is a surface conformation, which again works on the basis of the same van der Waals radii that I was talking previously. But here, instead of mesh conformation, you see the entire structure being covered. And based on the coloring scheme that we have, we are seeing the entire protein being colored based on different atoms. Like for instance, the coloring scheme that we see right now is green for carbon atoms, red for the oxygen atoms, and blue for nitrogen atoms. Based on this particular coloring scheme, you can see this particular visualization state. Now, based on what we are trying to address, for example, if you are looking at a binding pocket in my protein structure, then we go ahead with this particular conformation that you are seeing, because this helps us identify all the grooves that are present in the structure, all the pockets that are present in my protein structure, and as well as all the holes that are present in my protein structure can easily be visualized using the surface conformation. So based on this particular requirement, we go ahead by visualizing our protein molecules or any biomacromolecule in based on the visualization state we want. For instance, if you want to see the protein using their secondary structure conformation, then we would go ahead by the cartoon structure where we would be able to see various secondary structure, like for instance, the beta strand as well as the alpha helices structure of this protein. We would be able to visualize these secondary structures using this particular visualization state called a cartoon. Now, I think we have visualized mostly what are the different visualization state in which we can load our protein molecule. And if we want to hide the same corresponding visualization state, how do we go about hiding that particular visualization state using the hide button? So we have you explored both of these show and hide buttons. Now, apart from different visualization state, we can also see various other functions, the functional parameters that can be performed using these two buttons, the show and the hide. For instance, apart from the various visualizations, we can also visualize various properties like, for instance, the disulfide bonds. If there are any disulfide bonds that are present here in this particular structure, I can also try to visualize them. Like for instance, if I click on disulfide and click them on the lines conformation, you can see that here a particular line has been created which denotes the disulfide bond that you can see. If you would want me to show this in particular, let me hide all the cartoons. And you can see that a particular disulfide bond between two particular residues has been shown here. So this is another use that can be exploited using the show button. And again, I can use the same hide button if I would want to hide various other parameters that we are trying to see using the show button. Now, if you're going deeper into what are the different operations that can be performed using the visualization state, before which let me try to show the cartoon representation, we can use this action button. Now, action button helps us to zoom in and zoom out into the macromolecule. It helps us find any particular interactions that can be seen within this protein that I'm visualizing. It can help us to remove any particular atom, like for instance, if I would want to remove the water atoms, or if I would want to remove any particular residues that are present in this protein structure, we can go ahead by using the action tab that you can see here. And it also helps us mask any particular residues from the operations that we perform, or it also helps us compute any particular parameters, like for example, the distances, like for instances, the electrostatic calculation that we would want to perform, we can do that using the action tab. So we have various actions. Now, in order to explore only the most important actions that we would mostly be exploiting in order for our day-to-day studies, I will try to focus predominantly on some of the parameters that you see here. Now, as an example, we have taken the 3UCB structure. We have loaded this, which we have downloaded from the PDB database. Now, in the structure, if you see carefully, apart from the protein secondary structure, you also see a lot of red color star marks, right? I think you can see all these red color star marks. These are nothing but the water molecules that are being present in my structural files. If you all remember, when we were talking about the file, the coordinate file, from a text editor point of view, if we are looking at, sorry, was someone trying to talk? Okay, I assume that was nothing. So if I'm opening the same coordinate file using the text editor, if you all remember, I was mentioning about how different key terms have been used for representing different atoms that are present in my structural molecule. For example, every atom of the protein is being represented using this key term called the atom. So for now, for our better understanding, let us delete all the particular supplementary information that are seen in the header of this file. Now we have all the structural information that are being represented to show particularly that this is the atom of the protein. Now, if you scroll down, if you all remember, I had said earlier that apart from my protein structure, we also have the other key term called the heteroatom, which is represented as the het atom keyword here in this text file. So now this heteroatom helps not just show different secondary atoms that are present in my molecule, but it also helps us identify what is the particular molecule that is present in the visualization state. Now, for example, since my atom keyword ends here, this is exactly where my protein ends. And since our protein ends here, we have a key term called the ter, which stands for termination. So this is where one particular biomacromolecule terminates. Now, after this, we have a different keyword called the heteroatom. Now, we have different heteroatoms being seen here. And if you see particularly in case of atom, which represents the protein biomolecule, we see in case of the residues, we have different residues being represented. For example, you can see arginine being represented as ARG. You see lysine residues. You see isoleucine residues. You see glutamate residues. All of these being represented using a key term. Now, if we scroll down, when we see particularly in case of the heteroatom, we have different keywords here. Like for example, here what you see is EOH. EOH stands for ethanol, E stands for ethane, and OH stands for alcohol. So ethanol is the ligand atom that is present as the heteroatom, And if you scroll a little down, you see HOH, which stands for water molecules. So water molecules as well as the ethanol that is being present in my protein structural file are being represented here as heteroatoms. Now, how do I see this using PyMol? So let me close the text editor. I go back to PyMol, and I was saying previously, if I go to the display option, I have a particular visualization state for the sequence. Now, when I click on the sequence, you see the entire sequence, which constitutes all the components of the atom and the heteroatom being represented here. All the residue numbers from a particular protein are being numbered here, and all the water molecules that I see here as asterisk or the star marked conformations are being shown here using the respective colors. Now, in general, water molecules are always represented using zeros that are being shown in red color. And if I scroll again to the end, I can again see the same visualization state. So as I go on selecting this entire thing, so I'm selecting this particular conformation of water using my mouse, using the left button of my mouse. I have selected all of them. And you can see that each of the water molecule have now been selected. Let me deselect them so that you see how the changes are happening in the PyMol window. As I go on selecting, each of the water molecule are getting selected. So as I was saying, action tab helps us explore various options. Like for example, if I would want to particularly delete any of the atom, then I would go to this particular operation called the remove atoms. Now, whatever has been selected using the selection command, now if I click on remove atoms, this helps in removing all the atoms. Now you can see in the sequence view, we do not have the atom anymore and we are left with the protein alone. And again, as we scroll to the end of the sequence view, you see EOH that we were seeing in the file. So when I'm selecting this EOH, it particularly selects one particular atom here in the 3D space. Now, right now, we do not have any visualization because ethanol in this particular instance is the ligand, and we do not have a cartoon representation because cartoon representation is predominantly used for the proteins. So what we shall do now, once we have selected this particular ligand, which is the ethanol, we get a selection called the selected space here. So let me again show it for you, delete selection. As soon as I click ethanol, here there is a selection that is being created in my graphical user interface. So if I need to perform any particular action, or if I need to show or hide or change the color, particularly on the selection that I have made, which right now in this case is the ethanol. If we need to perform any of this activity, particularly on one particular atom that I have selected or any particular residue or a heteroatom I have selected, I would use this selection command that has been seen here. Now, if I would want to show this particular selection that is being made currently, which is the ethanol, I would go to show and I would try to show this in stick conformation. So that once I show this particular selection in stick, you can see here that there is ethanol being shown. So you have oxygen in one end and hydrogens being shown. So this is the particular ethanol that is being shown here is in the stick conformation. Again, if I would want to delete this, I would follow the same procedure where I would click on this so that a selection is getting activated. I would click on actions and I would go about to click on remove atoms, where now we have removed this ethanol, and you would see that ethanol component that was seen in the sequence has also disappeared. So now in the sequence view, all that we are left with is the protein residues that you can see all the amino acids are being shown using their key one letter representation. Now, in order to see different visualization state, now we clicked on sequence. So let us click again on sequence so that the sequence representation is lost. So sequence representation helps us to visualize the sequence of particular macromolecule which you have loaded. And now let me click again on the sequence so that you are able to see the sequence representation of this protein. Now, if I would want to see the sequence in different representations, for example, now what you see is one letter representation of each of the amino acid residues that are present in this protein structure. For example, the first residue in my protein is M, which stands for methionine. So it is represented using a single letter, which is M, that denotes methionine. But if I would want to represent this using a different representation, like for instance, amino acids also have a different representation, which is the 3-letter representation. If I want to show that particular representation, I would click on display. Here below the sequence option, I have a different sequence selection criteria called the sequence mode. So I would go here and I would click on residue names. So once I click on residue names, you can see that each of these residues are now shown using a different representation. Now the current representation that you see here is the 3-letter representation, which for example, in case of methionine is MET, MET. So this represents methionine residue, and glycine, methionine, serine. In this way, you see that each of the amino acid residues that are a part of this particular protein that has been loaded in the PyMol window have been shown using the 3-letter option. For example, if I would want to show all the particular atoms that are present in these residues, then I would click on the atomic names. And here, if you see each of these residues, for example, methionine, whose number is 295 in this particular structure, all the particular atoms, the nitrogen, the C alpha atom, the carbon atom, the oxygen, the C beta, and all the atoms that you see here for one particular residue are being shown. So if you would want to select and zoom in into one particular atom of this particular residue, you can use this option. So again, in order to see the sequence representation, we would use this sequence option. And if you would want to use a different representation of sequence, we would try to go about choosing all these different sequences. So this is one of the predominant function that you would want to see. And again, since we were talking about the mouse option that we have seen here, mouse is predominantly the main component that we use for controlling PyMol software, whether we would want to change the visualization state, whether we would want to rotate this particular macromolecule in the desired conformation that we want, whether we would want to zoom out or zoom in into this particular PyMol window that you see here. We predominantly use the mouse as our key operator. So now mouse can, in order to change the different functions that are being carried out by the left button, the right button, as well as the scroll button in a mouse, we can use this particular tab, which here in this menu is being shown here. But if you would want to see that in the GUI interface, you can also try to see that here. Now, predominantly, when we are talking about operations using mouse, we have two important key operations. The first one being the 3-button viewing, which by default is the default operation that is present for any mouse operation in PyMol window. And the second option that we have is the editing option. As you see here on the extreme right, if you follow my mouse cursor, you can see that it says 3-button editing mode that is being selected right now. If I click on that again, then I am able to easily toggle between 3-button viewing mode. So these are the two different predominant modes that we would use for controlling or communicating with PyMol window. So I can easily switch using my mouse using both of these particular modes. As you can see, I am easily able to toggle. In order to explore more mouse options, then we can go to this mouse window that you see here, and we can select also different functions. Like for example, you have 2-button editing mode as well as the 2-button viewing mode. Now let us explore what are the different functions that you can see using this particular mouse modes. Here, if you see below the visualization mode, you can see three different alphabets, the L, M, and R, as well as the V. So L stands for the left button that is seen in my mouse, M stands for the middle button, R stands for the right button, and wheel stands denotes the scroll button that is present, which we use for scrolling using our mouse. So now each of these particular buttons have been assigned one particular function. If you can see, ROTA, which represents rotation, has been given below my L button, whereas below my R button, we see a different function, which is move Z. And whereas
Johnson,
below
just
my middle
zoom in
button,
a bit.
we see
Just
a
zoom in
function
a bit.
called,
It's not visible, I guess.
okay,
Yeah, zoom in a little bit
okay,
because in the
probably
corner.
I'm not sure if we can,
Okay.
I'm not sure if I'll be able to zoom in into the PyMol window.
All right.
Probably
No
what I
problem.
can do is okay, what I can do is I can quickly take a screenshot of this particular GUI so that it's visible in a better way, and I would try to show it that way.
Okay. Yeah. Yeah.
I think if you can see right
Yes.
now, I have
Yes.
taken a snapshot of the particular GUI that I was trying to show. You can see three different mouse buttons that I was talking
Yeah.
previously, the left button, the middle button, as well as the right button. And you can see a particular function has been assigned to each of these buttons. For example, when I use the mouse in combination with the shift key that is present in my keyboard, I have a particular function. When I use the left button of my mouse in combination with the control key that is present in my keyboard, I have a different function that can be invoked. And at the same time, if I use the right button of my mouse in combination with the shift button in my keyboard, I have a different function. If I use the right button of my mouse with the control key that is present in my keyboard, I have different functions. So this way, we can explore multiple options in PyMol interface in order to communicate with PyMol using the limited three buttons that are present in my mouse as well as a few keys that are present in my keyboard. Now let me close this and let me go to something that we were seeing previously, which is the two button viewing. Now if I click on two button viewing visualization state, you can see here. Now let me take the screenshot again so that it's a zoomed in version for you all. Now, if you see here, when I clicked on the two button visualization mode, now you can see that the functions are being assigned only to the left button of the mouse, the right button of the mouse, whereas the middle button, which also helps in scrolling or does the wheel function, they have not been assigned any function because right now we have selected the two button visualization mode. So the crux of what I'm trying to make you all understand is we can control what are the different functions that we can carry out using different buttons that are present in my mouse. So this way I can assign a particular function to my mouse and I can do the desired functions using the PyMol window that I can see here. Now let me again go back to the default state, which is the three button visualization state. Now you see here by default we have the three button viewing, and on clicking on that we again have multiple modes being selected. So let me by default select the two important modes, one being the three button editing mode and three button viewing. Sorry, the three button viewing. So if I click on this particular space as you follow my mouse trackpad, so you can see that we have two different modes that you have selected, three button viewing mode and three button editing mode. So the particular mode that you see on the display is the mode that has been selected currently. So right now I'm in the three button editing mode. If I click on that again, that will help me toggle back to the three button viewing mode. Now, in three button viewing mode, what are the particular functions? Let me run you through the key functions that are present. So using my left button, I can control the rotation of this particular biomacromolecule using the PyMol window. You can see I'm clicking on the left button and I'm trying to rotate the particular biomolecule. So you can see that I'm able to rotate. Now, using the right button, I can help in zooming in and zooming out. So if I click the right button of the mouse and I drag it up or down, you can see that I'm able to zoom in and zoom out particularly. So the middle button, which helps in movement. So you can see the middle button here helps in the movement. So if I click on the middle button of the mouse and I move it to the left or the right, you can see that I'm able to move. So if I would want to particularly focus on this particular loop that you see here in the protein, what I would do is using the right button, I would zoom into that particular place. Now I would use my middle button to bring it to the center. I would again use my right button to zoom into that particular state, and using the left button, I can rotate this particular molecule such that I'm able to view only this particular loop. Now, if I would want to zoom in further so that only my loop is being seen throughout the window, I can zoom in further so that you can see only the loop confirmation that I would want it to zoom in. So now this is how we would use all the three buttons of our mouse to effectively do a particular function. So let me reiterate what I was trying to say. The left button of the mouse helps in the rotation of any macromolecule that we have selected. The middle button helps us in moving the particular protein to the left or the right or any particular direction. The right button helps us to zoom in and zoom out into the particular macromolecule that we have chosen. Now, on the other hand, as I was mentioning previously, the default mode that we have right now chosen is the three button viewing mode. If you can follow my mouse cursor, now let me click on three button viewing so that right now we have toggled into a different mode, which right now, as you can see, is the three button editing mode. So now the three button editing mode has different functions. For example, if I would want to move a particular atom, which again the left button helps in the rotation of the macromolecule, which is similar to that of the viewing mode. Whereas if I would want to do the same functions of moving and zooming in, I can do the same using the middle and the right button. But the difference between the viewing mode as well as the editing mode comes when I apply the secondary functions. Like for example, if you see here, we had discussed previously that we can also do secondary actions using the shift button of the keyboard as well as the control button. Now, when I click on the control button of this particular the keyboard and I use my mouse, the left button of my mouse, you can see how I'm able to alter the thing. But this particular function was not available in the viewing state. So that is the reason we have a different mode here for doing this particular function called the editing mode. Now, in what other ways does this editing mode helps us perform various functions? We'll be seeing that in the later part where we would take a real life example. We would go ahead by making publication quality images, and there I would be able to better represent what is the particular function that you can do using this three button editing. So mostly by now we have understood how to load a particular biomacromolecule into the PyMol window. We have understood how to visualize the sequence representation using the display sequence option where we visualize a particular sequence. Now again, let us go back to the sequence mode and select the residue names. Now we are seeing only the particular amino acid representations being shown here in the sequence mode. We learned how to do that. And using mouse, we learned how to control the visualization of a particular macromolecule that has been loaded. Like for example, rotating a macromolecule that is being loaded in the PyMol window, zooming in and zooming out of this particular macromolecule, as well as to move it onto any direction that is desired. So we have understood how to basically load as well as perform basic operations into this particular window. Now let us dive deeper into this particular visualization state and see how can we perform different functions. Now, before going into that, I would want to take you into the actions mode that we were seeing previously. So if you all remember previously, we were talking about the action mode where we can perform other options. For example, the find button that you see here, the modify, the present, the align buttons that you see here. And now let us explore each of these. Initially, as I was talking about selection, if I'm selecting a particular residue, now for instance, for your understanding, we are selecting two residues here. And as well as select two different residues, you can see a selection is being created here. Apart from the selection that you can see structurally, you also see a new tab that is being created here by the name selection. So we can perform any particular desired functions on this particular selection because have once we create a selection, a new tab has been created where if we want to show or hide or give a coloring pattern, we can perform it on the entire molecule or just the selection. Maybe if this was not visible, I can take a screenshot again and I can show the zoomed in version to you all. So if you see here, this is what the GUI looks like. If I try to blow up the size of this, so 3UCV is the whole molecule that was being loaded into the PyMol window. So each particular object or any particular macromolecule or any selection that is being made in a particular macromolecule is being given as a new column, the new row that you see here. For example, the entire molecule that was being loaded here is being given a row called 3UCV where I can perform any actions. I can show or hide a particular visualization state, or I can label, or I can specifically color it. Now, if I select any particular residues, maybe that could be an entire chain, or that could be specifically two or three residues, or that could be maybe 10 or 15 stretch of residues. If I perform any particular selection, again, the same actions that were performed on the entire protein can also be performed only on this particular selection. For example, if I want the entire protein to remain green color, but I want only the selected region to have a different color, I can go to the color button that is for this particular selection and I can perform the particular function. So this is how the GUI looks. Now again, let us come back to where we were. We have particularly selected two different residues, as you can see here. Let me select them again so that it is understandable to everyone. I am selecting residue valine and residue glutamine here. Two different residues have been selected. And as I select this particular residues, now we have a new row that is being selected, that is being created called the selection menu. Now, if I would want to particularly change only the color of these two particular residues, then I would go to color button in this particular selection row. And I would, for example, if I would wish to give it a red color, I would click on red. So that if I can zoom in and see particularly, now you can see the region of this protein where those two particular residues were being selected, we have a different coloring pattern now. Whereas the rest of the protein still has the same color because we have particularly changed the color only for this selection. Whereas now if I would want to change the color for the entire protein, I would go to this particular tab, this particular row, which denotes the entire protein. I would click on the color button and I would click on red. So you can see that the color of this entire protein has now been changed to red. Now, if I would want to particularly show these two residues that were being previously selected, these two residues in the stick conformation, I would go to the selection tab and I would click on show. And I was previously saying that we had different visualization state. And if I would want to see them in the stick conformation, I would click on sticks and you can see that those two particular residues, which is valine and glutamine that were being selected previously, are now being shown in the stick confirmation. In order to now differentiate this so that it is visible to you all in a different way, in a distinct manner, I would also wish to change the color. Now I would want to change this to white color. So I'm selecting these two particular residues and I'm changing the color to white. Now, if you see the region where these two residues are present, we can see that it has taken a white color, whereas the whole protein now still retains a red color. Now again, let me try three different conformations for you, different combinations so that you understand how it works. Now let me change the color of the entire protein to yellow. Now you see that the entire protein is being changed to yellow. But if I want to select the color, change the color of only these two particular selected residues, if I want to change them to blue color, if I click on the blue color, so you can see only these two particular residues have been changed to blue color. As you would start using PyMol, as you would slowly start getting hands-on on your own using mouse, you would start slowly gaining speed and you'll be able to quickly zoom in. You'll be able to navigate through these multiple options. You'll be able to move and rotate this particular molecule. So it's just a matter of time. You would be able to learn it over a period of time. So now we have understood how do we do a particular selection and how do we go about performing any particular function. Now, some of the functions that we saw was to show a particular conformation. Now so far we have tried to visualize this in stick conformation. Now, what we shall try to do is if I would want to show these two residues in another visualization conformation, like for example, now if I would want to show this as a mesh, now you can see that, okay, I have not selected them yet. So let me select these two particular residues again. And then if I would want to show them using the mesh conformation, you can see that these two particular residues have been shown particularly using the mesh conformation that you can see here. Whereas the rest of the protein is still in the cartoon conformation. Again, if I would want to show the rest of the protein, the whole protein in the mesh conformation, I would go to the row where it corresponds to the whole protein. I would click on show and I would click on the mesh conformation so that the whole protein now is being visualized using the mesh conformation. I hope so far all the visualization state, how to choose a particular visualization state, how to control our macromolecule, be it rotating it, be it moving it in the required orientation, or to zoom in and zoom out, all these particular functions are clear so far. So now let us dive in again deeper. Now, but before we dive in into particular functions, I would want to introduce you to a directory that is being given to us using PyMol. So this was the home window of PyMol that we had seen previously, using which we downloaded the software, and using PDB database, we downloaded the particular structure, whichever we wanted to visualize in our PyMol window. Now we are going to a different window, which is called the PyMol Wiki. PyMol Wiki is a short representation for the PyMol Wikipedia where we would be able to find different actions for my particular interaction with the PyMol window. Now, for example, let me just search. You might not be able to understand this. Let me, for example, search sphere. Sphere is one of the representation which we are seeing in the PyMol window. Now, when I type sphere, all the actions that are concerned with sphere are being shown in my PyMol wiki. If I would want to perform a particular action on the sphere representation of my in my PyMol window, I would type sphere in the PyMol wiki page, and I would be getting different actions that I would be able to perform. For example, what you see here as sphere transparency, I can zoom in a bit. So what you see here as sphere transparency is the particular function with which you can control the transparency of the spheres that are being visible in the PyMol window. Or for example, if you would want to control the quality of the spheres that are loaded in the PyMol window, you would choose this particular function. Or if you would want to particularly change the size of the sphere that is being loaded in your PyMol window, you can click on the sphere scale that is being seen. Now, how are we going to use each of these functions using PyMol wiki into the PyMol command that we are going to run? Let us now start understanding one by one. Let me take the example, the same example that we were trying to see initially. Let us open the 3 UCV structure which we were seeing previously using PyMol. So here you can see that we have loaded the same structure in PyMol again. Let me make it the full screen, and you can see it is the same initial structure that we have loaded. And like previously, what was done, we clicked on display, we went to sequence where we were able to visualize water that is present as well as the ethanol that is being present. So now we can go about deleting this by selecting them. This is one procedure where we can manually delete all the waters that are being present. If not, we can also do it using a single click where I was previously mentioning that we have multiple functions in the action button. So I click on the action button, and if you go down, so you have a particular function here called remove waters. So if I click on remove waters, using a single click, I'm able to remove all the water molecules. So in order to not select all the water molecules manually, like for instance, if you have a bigger protein molecule, bigger protein coordinate file where we have a lot of water molecules and manually selecting all of them would be a tedious job. We would simply go to the actions menu and we would click on remove waters. That would help us remove all the water molecules in a single go. Now we would scroll the sequence view to the rightmost. So we can see ethanol that is still present here. So we can click on ethanol, actions, remove atoms like we did previously. So you can see that the ethanol atom is now getting removed from my PyMol window. So as I was mentioning previously, how can we use PyMol wiki in order to perform specific activities in my PyMol window? So if you remember previously, I was talking about various visualization states. So I click on show and I click on this particular visualization state right now called the spheres. So if I click on spheres, you can see that now I have loaded a different visualization state, which is the spheres. So all the atoms that are present in my protein structure are now loaded as spheres. Now I again use my mouse buttons to control, to navigate to them, to zoom in and to rotate them. And again, the same color scheme that we had seen previously, all the oxygen atoms are represented using yellow color, carbons are represented using green, and nitrogen atoms are represented using blue atoms. So now we can see all the atoms in this particular protein are now being shown in the sphere conformation. So what now since this is very clumsy, if I would want to make you understand how does each of these sphere conformation work, what I would do is I would hide all these spheres right now. Let us keep this entire thing in the cartoon structure itself. Now, before going to do that particular function, let me introduce you to a selection criteria. Now, since we were previously talking about selecting, now the default selection criteria for any visualization state in PyMol would be a residue. So this you can see here in the extreme right. Let me take a screenshot and show a blown up version of this to you all. I hope now you are able to see the window that we were previously seeing. if you see, the selection criteria has been set to residues. Now, so residues is the default selection criteria that is being seen in a PyMol window. For example, if I click on any particular residue in the structure, it is one particular residue that gets selected. So which residue? Here you can see it in the sequence view. One particular residue, the Q is being selected. So glutamine is being selected using the selection criteria. Or if I would want to select it using the sequence, if I would want to select this particular valine residue, I would click on valine and the entire valine residue has been selected here. If I would want to show that to you, I would click on show and I would click on sticks. So if you see the stick conformation for valine residue, the entire valine residue has been selected because this happens because the default selection criteria has been set to residues. Now we can change this to any particular selection criteria. So if we go on clicking on residue, so we can see that we are able to see different selection criteria. Now to name a few, now we have the chains, which is the default selection criteria. Now we have segments. Now we have objects. Now we have molecules. Now we have C alpha atoms, right? So now what I'm trying to do, let me show all of the protein using stick conformation. I think now you are able to see the whole protein in stick conformation. Probably what I can do is hide the cartoon conformation for the whole protein. So if I zoom in into one particular residue, now let us dive in and zoom in into one particular residue. Now you can see that we are focusing on this particular residue. Now, if my selection criteria is set to residues, when I would click on any atom of this residue, you can see that the whole residue gets selected. For example, when I would try to color the entire thing, if I would change the color to yellow, you can see that the whole residue is being changed to yellow because the selection criteria is being set to residues. Now, if I would want to perform a function particularly on one particular atom, then I would change this selection criteria to atom. If you can see, I keep clicking till I come to this atom selection criteria. So now that means if I click on any particular atom, it is only that particular atom that is being selected. For example, if I would have the residues being selected, now you see that the selection criteria is residues. So if I click on any region of this particular residue, all the atoms of a particular residue are being selected. So if I try to show them as spheres, you can see all the particular atoms that are present in the residue are being shown using sphere conformation. Whereas now let me hide the sphere view and I change the selection criteria to atoms. Now once I change the selection criteria to atoms, I'm selecting only a particular atom. Now you see when I click on any region, it is selecting the atom that is closest to the region where I selected. Now when I try to show spheres, the sphere visualization state, you can see that it is only one particular atom that is being selected because the selection criteria was being set to atoms. Now this is the visualization that you see is a particular residue, one of whose atom has been set to be seen in the sphere conformation. Now let us go back to PyMol Wiki. If you all remember, we were trying to see what are the different functions that can be performed on a sphere. Like for example, here we have sphere scale, sphere quality, sphere transparency. Let us specifically go to sphere scale. Now, what did I tell previously regarding sphere scale? Sphere scale is the criteria or a function which helps us to alter the size of a sphere that can be seen in the PyMol visualization state. Now, you see in this particular window, you see a particular size of sphere that is being displayed. Now, again, before going into how we can use the commands from PyMol wiki into PyMol window in order to perform various functions, let me introduce you to particular command line that is present in the PyMol window. Initially, the region that we were talking, the right side console that we were primarily using in order to perform various functions is called the GUI, as I previously mentioned, which is the graphical user interface. But if you would want to specifically communicate with PyMol using commands, using particular commands where you would use certain keywords in order to say PyMol that this is a particular function that you would want to do, you would use this particular space or on the left hand side. If you can follow my mouse cursor, this is the region where you can type. For example, I would say that my name is Johnson. So whatever is being typed right now into the PyMol window is being taken in as a text into the PyMol command space. So this is the command input area where you can type in the command you want. So if you would now come back to the PyMol wiki page where we were trying to see if we can perform any functions, particularly by taking the help of PyMol wiki. Now, particularly, we are interested in understanding the sphere scale, which is the size of the sphere that we are seeing in the PyMol window. Here you can see one particular command, which is the get sphere scale. So here the explanation that is given for this command is you can print the current value for sphere scale. That means if you would want to understand what is the current size that is seen in your sphere currently in the PyMol window can be understood using this command called the get sphere scale. So let me copy this command. I am copying this command. So I would paste this particularly in my so I would paste this particularly in my PyMol window. And you could see I'm pasting it in the command prompt, which I was saying previously. I have pasted the particular command, which I copied from PyMol wiki, and I press enter. If you see this command prompt returns me with the value. If you see here, it says the sphere scale is 1.0. That means the size of the sphere is 1. I can alter the size proportions here. So if you see, once I type the command that I typed was get sphere scale, and PyMol returns me with a value called 1. That means the current size of this sphere is 1. Now, is there an option where I can change this particular value so that I can alter? So on a reverse mechanism, is there a way where I can set this particular value so that I can using a reverse mechanism, I can change the size of this particular sphere. So let us go back to the same PyMol wiki page that we are seeing. And if you can see here, you can see that there is this particular command called set sphere scale. So here you would be able to particularly adjust the size of any particular sphere that is being selected. Let me copy this command now. And I am going back to the PyMol window that was being opened. So I am pasting this command here. You can see the command that was being copied from PyMol wiki page has been pasted here. The command that you can see here is set sphere scale. Now the initial scale that was being set previously was 1. So let me see if I can particularly set 0.5 as my state, and on what particular residue on what particular selection am I going to apply this. So the selection here is called this selection tab, so which is represented using SELE, which stands for selection. So let me type that particularly here, SELE, and once I enter, so okay, sorry, I forgot to put a comma here. So once you put a comma here, and sorry about this, let me go back to PyMol wiki page. Okay, here you can see the command where we can specifically change the size of the sphere which we want to control, or here there's a better representation. This command helps us to get the particular size of the sphere, whereas the changing of a sphere scale can be done using this command. So I'm copying this command as it is. I will now go to the PyMol wiki, the PyMol window, and I'm going to the command line prompt of the PyMol window. I'm pasting the entire command here. So what is the particular size that I want? Let me set the size to 0.5. And what is the selection that is particularly being seen here is select. So let me type select over here, selection, and uh, just give me a minute. So there's a small glitch with this program. So let's open the PyMol window again. Once again, right click. Is there an option where I can erase all the marks that are being present on the screen? Yasha, is there an option? Can you please erase all the lines that are seen on the screen?
Uh, I can't erase it.
Okay.
Yeah, I can.
Okay, that's not an issue. That's not an issue. Let's just go ahead with what we're performing. So I'm opening it again using PyMol window. So open using PyMol. So we are opening pre-UV structure again using PyMol window. So I'll make a full screen. Now, as the selection criteria that we were previously talking now, which is by default set to residue, residues. Let me change this particularly to atoms. Now we have changed it to atom. Let us particularly go and select one particular residue from the display option. So I have the sequence. I'm selecting one particular residue, which now, for example, let us go to the end and select one particular residue. We have chosen one particular residue, so we will show that using a stick confirmation, show stick. So now we are able to see this particular residue that was being selected using the stick confirmation. Now, since we have chosen the atom representation in the selection criteria, I will click on one particular atom, which is the nitrogen atom. I'm selecting this nitrogen atom and I'll click on show and I'm selecting the sphere. So you can see that right now nitrogen atom of that particular residue has been changed to sphere selection criteria. And now let me go back to PyMol Wiki page, where what I try to do is take the same command which we had taken previously. So get sphere scale is the command that I would want, that I'm interested in. I'm pasting this command here again, get sphere scale. So I'll press enter and you can see, like we saw previously, it is saying that the default size of this particular sphere is 1. Now, if I would want to alter the size of this particular sphere, again, I would choose this particular command, which is set sphere scale size selection. I'm copying this command. I'll paste it into the command prompt. And you can see here this asks us for a particular size. And now let me specify the size to be 0.5 and I'm selecting Sele. And if you see now, I particularly tried to see if I can change the size of my protein, the sphere, which was initially 1 to 0.5. As I changed it to 0.5, you can see that the size of the sphere has completely reduced. Now, probably, let's say I would want to decrease the size of the sphere to 0.25. That would make the size of the sphere into half furthermore. So if I click enter, you can see that the size of the sphere has completely reduced. So this is how you can particularly make use of PyMol wiki page where, for example, each of these functions that you would want to perform in PyMol, you have different criteria that you can see using this. Now, let me go back. If I'm talking about the changing the size of a sphere in PyMol window, I can use this function called the sphere scale. If I would want to similarly change the quality of a sphere, or if I would want to change the transparency of my sphere, I would use various functions that are being shown here using the PyMol page. So this is how I would use the commands that are given to me using the PyMol Wikipedia page. So I can use those commands, specifically type them in the PyMol window, and I can see if I can do a particular function. Now, so this was one explanation how we can effectively use PyMol Wiki to specifically type in commands to do a particular function. Now, apart from that, using the graphical user interface itself, what are different other functions that we can perform? So let me take you through some of the key functions that can be performed. So the first function that I would want to show you is the transparency function. Like for example, let us again clear everything. So I will open a new PyMol window where I'm trying to load the same 3UZV structure, open, and I would open the same, give me a minute. So let us open the same structure which we have been seeing previously. And again, the same set of functions that we were previously performing, display sequence. So now this shows us the whole sequence of the protein as well as the heteroatoms. Let us delete all the waters that are present here. So I'll click on remove waters. This by default removes all the water molecules from my visualization state. Now, transparency is one particular function with which we can control to what extent should my macromolecule or a different visualization state of my macromolecule should have the transparency. For example, now if I would want to control the transparency, I can control specifically the transparency of a surface. I can control the transparency of a sphere, control the transparency of cartoon, or particularly for the sphere. So what I can try to do is now let me go to settings, click on transparency. Now, since we have the cartoon representation, which is seen on the screen, I will click on cartoon and now we have different percentages to which we need the transparency to be set. For example, right now by default, it is the transparency is set to off. So by default, we see the entire cartoon representation as an opaque image. So let us click on 50% transparency. You can see that there has been transparency activated on this particular protein structure. If I would want to further increase the transparency of the particular cartoon representation that you see, I would click on transparency. Again, I would go to cartoon and I would increase the transparency. So you can see that particularly now, the cartoon representation has been set to 80% transparency. Now, in order to understand this better, now let us try to load two different confirmations, two different visualization states. Let us turn off the transparency for cartoon. So we have turned it off so that now the entire cartoon is in opaque form. Let us also try to show a different visualization state, which is sticks. So now you see that particularly both sticks as well as cartoon are being shown. So what I'm trying to say is that we can have multiple visualization states that are being loaded simultaneously onto the PyMol window. Similarly, now what I can do is I can hide the sticks confirmation. I can show the surface confirmation as well. So if I click on surface confirmation, so right now what you see is we have two different confirmation. One is the cartoon, one is the surface. However, now in this window, if you're seeing, we are able to see only the surface confirmation because the cartoon structure is not visible because it is beneath the surface confirmation. So how do we go about seeing both simultaneously? By now you would have figured out that if we can adjust the transparency of the surface, that is covering the cartoon structure, we would be able to see both the cartoon as well as the surface confirmation. So how do we do that? Let us go to settings. I click on transparency and I would change the transparency of the surface to 80%. So now if you're seeing this properly, if you see it carefully, you can see that the transparency of the surface is being set to 80%. That means you're effectively able to see both the surface as well as the cartoon representation at the same time. Now, if I would want to make this a little more opaque, then I would change the transparency of surface to 60%. You can see that the transparency has reduced. Now you can effectively see both the surface as well as the cartoon representation of both these things. Now, how do we use this effectively when we are talking in terms of publication? Let me take a different example and show this to you. So let me go to file manager. So I have a different file here, a protein, a PDB format file, a coordinate file. So let us load that in PyMol. So here, what we are seeing is two different proteins that are interacting with each other. You can see here on top, when I see the sequence view, you can see A. A stands for A chain, which is the A chain that is interacting with another protein, which has been represented as the B chain. So I was talking previously about selection criteria to particularly select either a residue or the atom. Similarly, we can also select the entire chain. Now, since we spoke about chain A as well as chain B here, let us see if we can perform the activity on a particular chain as a whole using the selection criteria. Let us go back to the selection criteria on the extreme right here. So I click on residues, which is by default the selection criteria. Now, as I click once, I see what is called chains. I think you are able to see this font over here, which says chains. So now I have set the selection criteria to change. That means I select any particular residue of a particular chain, the entire chain is getting selected. You can see that the entire A chain has been selected. If I would now want to change the color of the entire A chain, I would click on color, I would click on red. Now you see that the color of the entire A chain has been changed to red. You can see that, right? And now, if I would want to particularly change the color of my chain B to particularly blue, let's say I would click on any residue of chain B. Since the selection criteria has been set to change, the entire chain is now getting selected. So I would go to coloring button of this particular selection. I would click on color. I would click on blue, and now you'd see that the entire chain B has been set to blue color. Now, probably let's talk about the background color. Probably it's a little gloomy because we have the background color being set to black. So there is this particular function under the display tab. If we go to background, we can change the color to white. Now, if you see the background color has been set to white, the same can also be done. Now, this change of the background color was done using GUI. Can that same be done using command line as well? There's a particular command here called set BG color to black, let's say. Sorry, set BG color and set BG to black. Uh, Sorry, let us see how we can change that using PyMol Wiki. If we want to change the background color, let us type background color in PyMol Wiki and see if we can get any commands by which we can change the background color. Okay, you can see here the background color. There has been a command here in PyMol Wiki page. We particularly click on that. And so here the background color can be changed using this particular command. So I'm selecting this command, I copy this and I paste it in the PyMol window. As you see here, I'm selecting the PyMol window. I'm going to particularly the command line prompt. I type in the command and then I type the color that I want to type. So once I type black, which is the color to which I would want to change my background, you can see that the entire background color is being changed to. Let me show you a different page, which will there's a particular page called the PDB sum, which helps us understand the interactions between two different biomacromolecules. So I would particularly click on the protein-protein interaction. Although this is not a part of the module, I'm trying to show how can publication quality images be made. So if you see here, different residues of chain A and different residues of chain B, we see different type of interactions between both these chains of residues between the two different proteins. So you can see here there is a legend for what are the different colored interactions. So all the hydrogen bonds are being shown using blue color stick-like structures. So if you see here, the interaction between arginine 86 of chain A and glutamine 116 of chain B are having a hydrogen bond between each other. Now let us see if we would want to replicate this into a publication, how would we go about doing this? So let us go back to the PyMol structure that we have. We are selecting this particular structure. Now we are interested in seeing arginine 86 of chain A. So now we are in chain A. Let us go to arginine 86. You can see here based on the numbering, you have arginine. Now since we are in the chain selection criteria, the entire chain gets selected. So let us go back to the residue here on the right extreme end. You go back to the residue selection criteria. Now you would be able to select particular residues. Now we are interested in arginine 86, so we click on arginine 86 particularly, and we are seeing if there is an interaction between arginine 86 and glutamine 11. So glutamine 11 of chain B. So we go to chain B and we would specifically choose glutamine 11, which is here probably. Uh, Chain B, glutamine 11, but we have a different ratio here. So let us zoom in now and show these two residues that we have selected using show and sticks. Now you can see that both of the residues have been shown here using the stick confirmation. Probably the residues have not got selected. So let me close this entire file. Let us open the file fresh. So I'm opening this file called interaction, where we are trying to see the interaction between two different chains. So we have chain A as well as chain B. We are going to specifically see the sequence view, and chain A particularly we are selecting arginine 86, where we go to arginine 86 of chain A, and at the same time we are also selecting glutamine 116 of chain B. We go to glutamine 116, which is over here 11, 12, 13, 14, 15, 16. We have chosen glutamine 116. Now both of these residues have been selected, before which let us try to change the color of the entire background by display background to white. And we again select those two particular residues, arginine 86. So we have selected both of these residues. Now let us click on this particular selection. Let us choose the color to red. So you can see both of these residues are now being shown in red. Now, in order to understand the molecular interaction, let us try to show them using stick conformation. Now we have tried to show both of these residues using stick conformation. Let me try to increase the size of the window. So you can see both these residues have been enlarged as the stick view. Now, previously I was mentioning about this label command where we would be able to particularly visualize the labels of particular residues. Now let me click on the residues. Now you can see that a labeling has been done. If I could zoom in carefully, you can see here that there has been a label for both of these residues that are being created. Now, initially I was telling that editing mode can be used for various purposes. Now, one of the purpose which we use the editing mode is to, in order to move these labels from one place to other place, let us click on the edit. That using the editing mode, I'm specifically able to move this particular label, and at the same time I can move the label for another residue as well. So now here you can see what generally we see in publications. You see the two different residues whose interactions we need to study have been shown using a different color and using a different conformation. We have labeled them, and in order to find the interactions between them, we go to actions and we go to find. And particularly since we are seeing the hydrogen bond between these two particular residues, we would click on polar contacts within selection, and you can see that the hydrogen bond between these two residues has been shown using yellow dotted line. So this is the particular hydrogen bond that was being seen previously in this particular prediction that was being made online using the software named PDB-Sum. You can see that there is a hydrogen bond between these two particular residues, and we have tried to show that using PyMol. So you can see that these two particular atoms are being connected using a hydrogen bond. Now, in order to calculate what is the distance between these two particular atoms, what we do, we click on the wizard options, we click on measurement, and we select this particular atom as well as the other sister atom. And now you see that the distance between these two particular atoms where we see a hydrogen bond is 2.2 angstroms. And now if I would want to change the position of this, again, I would go back to the editing mode. I would click on control, and if I move this, you see that I'm able to move the particular distance that is being labeled. And now in order to make this, to make our focus particularly on this particular interaction, it would be better like in publications if we are able to decrease the transparency of the cartoon structure. So like we saw previously, we go to settings, we go to transparency options, and we click on the transparency of the cartoon, we set it to 60%. So now you can easily see that the cartoon has been made transparent, and particularly the focus has been shifted on the particular interactions that is between arginine 86 of one chain with arginine 118 of the 116 of the other chain, and what is the interaction between them. So this is how publication quality images based on a particular necessities, like for instance, checking the interaction or to perform any other function can be done using PyMol. Now, the last thing that I would want to cover is we have seen how to perform different functions using PyMol GUI. We have seen how to perform different functions using PyMol command line. At the same time, we have seen how to use PyMol Wiki to perform different functions onto the PyMol window. Now, can we do all this using any script? Because right now, most of the programming language based setup helps us to write the particular code and perform all these functions without manually having to do them on our own. Can we do that? Yes, that can be done using PyMol script. As I was previously talking about something called the PML file, which is by default the PyMol script is nothing but a bunch of commands that we enter in the PyMol screen. So that can be used for performing certain functions. Like for example, in this particular PyMol window, if you see, I have three different commands. Fetch 3UZV is the command that helps in bringing all the 3UZV structure from the PDB database. Hide cartoon, as we saw previously, helps in hiding the cartoon representation in the file. Show surface helps us to show the surface representation of this particular file that we have fetched from PDB database. How can we go about using this script to perform all these functions using a single click? Let us quickly see using PyMol window. So we'll open PyMol window. And if you would see here, we have the file menu onto which we have a particular option called run script. So this run script helps us to load a particular script file which has all the set of commands we would want to run instead of having to run them manually. So I click on run script. Now I would have to select that particular script file which was saved locally in my device. I would go to that particular location. I would select that particular script file which I have saved. So once I load the script file, it helps in automatically running all the script that was being present. So if you see here, all the three functions that were being typed into my script file has been loaded. So the first command was fetch 3UZV, where it goes, the PyMol interface goes to PDB database. It collects this structure which has the PDB ID 3UZV. It hides the cartoon representation and it shows the surface representation. So whatever was done manually using multiple steps has been done using a single step by uploading all the commands into the script file. And you can see we have the final uh, representation of this structure in the surface conformation. So this is how we can also employ script for particularly doing any of the functions that we need to do using the PyMol window. And finally, if you would want to save these all the actions that you want to perform, there are two different ways of saving. Now, any action that was being performed in the window, like for instance, here let us perform two different functions. I go to action, I go to remove waters, I have removed the waters. I go to display, I change the background to white. Now the background color has been changed to white. And I would try to change the color of this entire protein to cyan. Now you see that the entire color has been changed to cyan. Now, I would wish to save the entire session. So whatever changes have been done to this particular file, if I would want to save them, I would click on file, I would save session. So this would save as a PyMol particular session file. So let me save this as session_1. So this has been saved as a particular session. So now if I would want to start from where I left at the previous point, I would open this session file using PyMol, and you can see that instead of opening from scratch, I'm opening from that particular point where I had stopped performing various functions of the PyMol window. So this is how I can save my session files into my local disk. And finally, there are a set of commands that I would use for particularly making publication quality images. So these are the set of commands which mostly various people try to employ in order to make publication quality images. By default, most of the publications would have a background white color. And the important functions we are trying to see here is ray command. So ray command specifically helps us to increase the photorealism. That is, it's a computational term where we try to increase the resolution of the image, which computationally is termed as the photorealism. We try to enhance the resolution of this image by giving a higher number. So using this, we can particularly save any particular resolution into a higher resolution for publication sake. And in order to save that locally into a machine, we can run this command where we can particularly give the location and the name of the file. Like for instance, if I would want to save my file, the final image in this particular folder, I would copy this path. I would replace the location using this particular path. So this is the path in which my file would get saved. If I would want to name my image, the figure into a file named session or maybe image. So this is the name with which I can save the particular file that I would want to. So these are the set of commands. Let me zoom in. So these are the set of commands with which we can save all the sessions or all the actions that we perform using PyMol window specifically into higher resolution images for publication quality. So I think this wraps up most of the hands-on part of the PyMol action window that we had been performing. So let me now particularly take you to some of the images that were being performed using this. So here I think now you would mostly be able to understand what are the different coloring schemes that is present. Let me zoom into one of the picture. So if you can see here, one of the image that has been taken from publication, you can see the different secondary structures have been given a different color. For example, the loops have been given a white color. The alpha helices have been given a gray color, whereas the beta strands have been given the black color. All the active site residues of my particular protein that are being seen here are being shown as red sticks and red mesh structures. Similarly, we can also display a particular protein in using the surface representation, which as you can see here, we have the entire protein being shown in the surface representation, and you can see the ligand which is bound to this particular protein is shown as the stick conformation. And the final example image that I would want to show, which has been performed using the same set of commands that was shown to previously. In this particular image, what the authors have tried to show is they want to show the particular interaction between the ligand and the residues of this particular protein. So the protein has been shown using gray color. The interacting residues of this particular protein are being shown using green color, and the ligand has been shown using the cyan color. And all the interactions, like we saw previously, have been shown using yellow dotted sticks, and the distances have been measured. So these are some of the example images that can be performed using all the hands-on that we saw previously in order to make publication quality images. So I will stop sharing my screen. If anyone has a doubt or anyone has queries, you can please go ahead and ask me.
Um, Thank you so much, Mr. Samuel. Your presentation was truly engaging. I truly appreciate it. And the best part was you started from the very fundamental concepts and gradually you was building upon them that made it exceptionally valuable. The best part, the great thing about your whole presentation also was that you have explained each and every options, each and every part of PyMol, giving every detail. So thank you so much. No doubt the viewers have gained substantial knowledge and familiarity with the capabilities of PyMol through their through your presentation. So we have few questions. We'll take few questions as we are running out of time, so we won't be able to address a lot of questions. But yes, we'll take few. So I want to invite audience to share their questions and queries pertaining to the discussed topic in the comment section. A few that we have got, out of which one is okay. So when once we have the structure ready with all the analysis and we want to put it for publication or something, then if we need to save that as an image in 300 dpi resolution, how can we do that using PyMol?
Okay, so
Yeah.
if you see, should I go ahead and share my screen? Probably I'll share my screen and quickly try to answer the question. I hope my screen is visible to all. If you're
Yeah.
seeing previously that we have this particular command where we try to export all the publication quality images to a particular DPI. So the entire gist of these commands that I had shown previously was to make publication quality images. So if you see the last command where we save it into a particular PNG file, which is the image format file, we have the command which specifies the DPI. So here we have set the DPI to 1000 because by default, a lot of publications, some publications have a DPI requirement of 300, some have a requirement of 600, some have a requirement of 1000. So if you would particularly want to save it in 300 DPI, you can change this command where you are setting the DPI to 300. So thereby the image that you're going to save using this particular set of commands would have a 300 DPI resolution.
All right.
I hope that's clear.
Yeah, definitely. The next question was when we have the structure and we want to know the energies of each and every residue, then how could we do that? If we want.
Okay. So now, if you're talking about energy, now PyMol has certain limitations. PyMol has been designed to understand certain things. Like for example, it helps us to visualize. It helps us to check the interactions between two different macromolecules. It helps us to visualize using different visualization states, like the stick, the cartoon. It also helps us understand the electrostatic potential of each of these particular residues. For example, which of the residues have a negative electronegative potential, which of them have a positive value for the electronegative potential. These kind of activities can be performed using PyMol. Whereas when we talk about energy, energy is a higher term which we cannot study using PyMol. So there is a hierarchy of softwares that are being used in order to study these things. For example, in order to answer this question, although this is not the scope of today's webinar, to answer that question, I would say that energy is always studied in terms of delta T, which is gift-free energy. So in order to get to gift-free energy, we have a lot of calculation criteria, we have a lot of online servers, or we perform an MD simulation study. So probably Neighborman is conducting a lot of webinars in the future, a lot of internship programs, which will cover these aspects where we perform MD simulation. So probably what we are trying to see in PyMol is only the visualization and understanding some primitive functions that can be done on the protein molecule. So if you want to study this particular function that we're talking, the energy, like for example, the gift-free energy, the delta G value, we would perform an MD simulation studies, after which we would study the energy status. So there are certain programs like essential dynamics, after which we do a free energy landscape. From free energy landscape, we would get a delta G value, which denotes the energy of that particular molecule. So that is beyond the scope of what PyMol can function.
Hi, Johnson. Thank you
Hello.
so much for explaining PyMol very nicely. I just want to know how, when we are exporting this image as PDB file, it is asking for, there are so many options available there. I mean, I couldn't understand that. When you see that export molecule as PDB, you can see -1, 1, 0, and enabled selection protein. I mean, it is showing so many options. So,
Yeah.
can you explain
Okay.
what it means?
Okay. Let me run you through that. So these are some of the parameters with which you would want to save a PDB file, because like I was mentioning previously, if you want to save a PDB file, so we mainly are talking, when we talk about any coordinate file, we are predominantly focusing on the coordinates, the X, Y, and Z coordinates. So when we save the states, there are multiple states in which we can save all these particular files. Because now if I would go into that, the detailing is huge, because we would have to talk about all the coordinates and what are the visualization states that can be saved. So by default, you can save all this to the default parameters. And I think when you click on export molecule, because when I previously I explained how to save a session, and I mentioned how to save this as a PNG. Now, if you would want to save the entire PDB, for example, you are creating a docking analysis and you would want to save this entire thing as a PDB. So I click on export molecule, and the state that has been given has to be set as -1. -1 is the default visualization state in which you save your PDB, such that the code